Welcome to this explainer. Today, we're stepping into a true biochemical masterclass. We are going to completely demystify the complex machinery of life by looking at exactly how proteins are built from scratch. Think of this as a lecture on the ultimate microscopic architecture that keeps you, and frankly, every living organism alive and functioning. Let's get into it.
All right, here's our syllabus for today. First, we'll meet the cell's workhorses. Then we'll break down the building blocks, amino acids. After that, we're looking at the 4 levels of folding, we'll compare fibrous and globular proteins, and finally, we'll see what happens when protein structure fails.
So, Part 1. Meet the cell's workhorses. Let's establish exactly why we should even care about these molecules in the first place.
Listen, proteins are absolutely the ultimate workhorses of your cells, taking on a massive, almost unbelievable variety of roles. I mean, they're physically building the structural tissues of your body, like the collagen in your bones and the keratin in your hair. But they're also acting as dynamic machines. Enzymes speed up metabolic reactions, like amylase breaking down starches when you eat. Transport proteins, like hemoglobin, carry oxygen through your blood. Antibodies actively tag invading viruses for destruction. And hormones act as complex chemical messengers, regulating things like your blood sugar. They literally do it all. Moving on to Part 2, amino acids, the building blocks. To understand how these incredibly diverse machines actually work, we've got to play the role of molecular architects and zoom all the way in on their basic pieces.
And here is the craziest part. Every single human protein, no matter how wildly complex its final function might be, is constructed from a standard toolkit of just 20 standard amino acids. Think about that for a second. Just 20 basic monomers are linked together in countless variations to create the massive diversity of proteins in your entire body.
So how do 20 pieces do so much? Well, every single amino acid shares the exact same basic backbone, a central carbon atom, an amino group, and a carboxyl group. But attached to that central carbon is something called the R group, or side chain. This R group is basically its personality. It alone dictates the amino acid's specific chemical behavior, determining whether it'll be hydrophobic and fear water, hydrophilic and love water, or carry a positive or negative charge.
Biochemists actually categorize these 20 building blocks based on how their specific R groups behave in a watery cellular environment. You've got the nonpolar amino acids, which are mostly made of hydrocarbons. They're hydrophobic, meaning when a protein folds, they tuck themselves away into the interior core just to escape the water. Then you have polar uncharged, along with charged basic and acidic amino acids. Because these possess electronegative atoms or full charges, they're hydrophilic. They absolutely love water and typically sit right on the outer surface of a folded protein, constantly interacting with their environment.
So, how do we snap these different personalities together? It happens through a process called dehydration synthesis. The carboxyl group of one amino acid reacts directly with the amino group of the next one. In doing so, it strips out and releases a molecule of water, H2O, and creates a remarkably strong covalent connection known as a peptide bond. By repeating this process over and over, the cell creates a long linear chain of amino acids called a polypeptide.
Let's look at Part 3, folding and the 4 structural levels. This is where we figure out how a flat string of amino acids turns into a 3D machine.
A protein simply cannot do its job as a flat polypeptide chain. To gain its functional abilities, it must fold through 4 distinct structural levels. If it fails to achieve this architecture, or if it loses its shape later on, it completely loses its ability to work.
It all kicks off with the primary structure. This is just the exact linear order of the amino acids linked together by those covalent peptide bonds we just talked about. It's essentially a biological sentence, and this highly specific sequence is determined directly by the genetic code written in your DNA.
Then, that primary chain naturally begins to fold into its secondary structure. Atoms right along the protein's backbone begin forming hydrogen bonds with each other. These hydrogen bonds force the once flat chain into highly regular, localized patterns. It's either going to coil up into a spiral shape known as an alpha helix, or it's going to fold back and forth into flat, parallel ribbons called beta-pleated sheets. These structures provide essential stability and flexibility to the growing protein.
Next up, we reach the tertiary structure, which is the full 3-dimensional folding of that single polypeptide chain.
This is where those unique R groups finally take center stage. Those water-faring hydrophobic groups rush to hide inside the core, while polar groups form hydrogen bonds on the outside. Plus, certain amino acids like cysteine have sulfhydryl groups that react with one another to form incredibly strong covalent disulfide bridges. These act like permanent molecular staples, aggressively locking that functional 3D shape right into place.
It's really an epic progression. We start with a primary beaded necklace of amino acids, advance into coiled alpha helices and folded beta sheets, and finally crumple together into a highly complex, dense, tertiary three-dimensional shape. And for some complex worker proteins, like the hemoglobin carrying oxygen in your blood right now, there's even a quaternary structure. That's where multiple fully folded chains associate together to form one massive functional unit.
Part 4. Fibrous versus globular proteins. Once they're fully folded into their ultimate 3D shapes, these macromolecules generally fall into two broad, very different, real-world camps.
On one hand, we have fibrous proteins. These are the strong, structural pillars of the biological world. They consist of long, rod-like molecules linked by strong intermolecular bonds. They're insoluble in water and mechanically tough. This is the collagen in your connective tissue and the keratin making up your hair and nails. On the other hand, we have globular proteins. These are the delicate, spherical worker bees. Because they're meant to operate inside the watery cell environment, they are water-soluble. Your enzymes, your circulating hormones, and your hemoglobin all fall right into this highly active globular category.
Now, because globular proteins are so highly specialized and spherical, they're incredibly sensitive to changes in their environment, particularly heat and pH. Take the common fried egg, for example. If you've ever cooked one, you've actually performed high-level biochemistry. The clear egg white is packed with a globular protein called albumin. When you apply the heat of a frying pan, you are violently disrupting the delicate hydrogen bonding between the polypeptide chains. The chains open up, tangle, and solidify. This permanent loss of 3D shape is called denaturation. You literally scrambled its structural integrity.
Which brings us to our final section, Part 5, when protein structure fails. What happens when the cellular assembly line makes a biological mistake before the folding even begins? While the architecture of a protein relies on incredibly specific chemical interactions, if your DNA has a mutation, it might instruct the cell to place the wrong amino acid into the primary sequence. Swapping out even a single amino acid brick, like changing a hydrophilic R group to a hydrophobic one, can cause the entire biological building to fold incorrectly and completely collapse.
Sickle cell anemia provides a dramatic, real-world example of this. It's a devastating disease caused entirely by the abnormal formation of the hemoglobin protein. Just a single, microscopic alteration in the primary sequence changes how the hemoglobin folds, causing a normal, healthy red blood cell to physically deform into a rigid, sickle shape. These deformed cells end up clogging blood veins and severely disrupting normal blood circulation throughout the entire body. A tiny structural error yields a massive biological catastrophe.
It really leaves you wondering, doesn't it? As we wrap up this explainer, the sheer precision required for life is staggering. If one single molecular typo out of hundreds of amino acids holds the power to alter your blood and health so profoundly, what other incredible secrets, strengths, and vulnerabilities are written in the thousands of unique biological sequences operating inside you right this second? Thank you for joining me for this masterclass. Keep wondering and keep learning.